Hello everyone, I am Audrey Denizot. I am a postdoctoral scholar at the Okinawa Institute of Science and Technology, and I'm working in the computational neuroscience unit led by Professor Eric de Schutter. And today I'm going to present to you my work on astrocytes and their calcium activity. More specifically, I will present to you the work that is in the paper that has been accepted in the ACM Nanocom 2021 proceedings. Here you have the list of all of the co-authors of the paper, so Activiry, Eric de Schutter, Claude Cali, and myself. And I would like to start by thanking Jari Hyttinen to have invited me and given me such a nice opportunity to share my work with you, as well as the conference organizers. So when we talk about information processing in the brain, the first things that come to our mind are neurons and their network. But neurons only account for actually half of our brain cells, the rest being called glial cells, which includes astrocytes, that are the cells that I am interested about. So astrocytes have many different roles in brain function, including being essential for its development and its homeostasis. But here we're going to focus on their role in information processing in the brain. So in this 3D reconstruction from electron microscopy from Henneberger and collaborators, you can see in light blue an astrocyte, and in gray and yellow, you can see some dendritic spines, and you can see the physical proximity between the astrocyte ramifications, also called processes and branchlets, to those spines. And indeed, astrocytes are opposed to synapses. But this is not just the physical interactions, and actually astrocytes communicate with neurons at synapses. How does that work? So on this video from Arizona and collaborators published last year, you can see in pink a dendrite, and in gray the morphology of the astrocyte resolved with super resolution microscopy, and superposed on it you can see calcium imaging from confocal microscopy. And what we can see is some hotspots of calcium activity just at the vicinity of dendritic spines. And this is how we measure activity and cellular communication in astrocytes. This is due to transient variations of the calcium concentration in the cell. And as you can see, it can be quite localized at the vicinity of synapses. And this is due to the fact that active neurons release neurotransmitters that activate receptors on the membrane of the astrocyte, which results in molecular reactions that lead to those calcium signals. And those signals are believed to enable the release by astrocytes of gliotransmitters, molecules that will modulate neural communication. And those calcium signals are essential for the molecular basis of, the cellular basis, of memory and learning, that is long-term potentiation. And if you inhibit astrocytic calcium signals in mice, it results in deficits in motor coordination as well as memory and learning, which shows that those astrocytic calcium signals seem essential for information processing in the brain, but the mechanisms responsible for those roles of astrocytic calcium signals in information processing remain poorly understood to date. And this is notably due to the fact that astrocytes are characterized by very complex morphology, often referred to as spongiform, as you can see here on this 3D reconstruction of astrocyte morphology from electron microscopy from Shigetomi and collaborators. Indeed, 75% of the cell volume consists in branchlets, processes that are so thin that they cannot be resolved by diffraction-limited microscopy, and others, except if using super resolution microscopy, are not resolved when doing experiments in live tissue. So this is a huge barrier limiting our understanding of astrocyte physiology. Notably due to the fact that those fine processes are the very site responsible for 80% of calcium signals in the astrocytes. And importantly, most of those signals are what we call compartmentalized. So that means that the signals are highly local and do not spread within the cell. And this is very interesting because one astrocyte in the hippocampus is believed to contact up to 100,000 synapses simultaneously. So those compartmentalized signals could allow one single astrocyte to manage many different functions and communications with different synapses at the same time without one impacting the other. However, in some cases, the signals do propagate in other branches, potentially impacting other synapses and sometimes even leading to what we call global responses, in which the whole astrocyte is characterized by increased calcium concentration. We don't understand the mechanisms that are responsible for those various spatio-temporal characteristics of calcium signals. And we also do not understand whether those diverse signals are associated with different functions from the astrocyte. And this is what we call the astrocytic calcium code. And for the moment, it's highly, it remains to be solved, basically. We still don't understand the roles of those calcium signals. So in this work, we want to better understand the factors that can influence those diverse calcium signals. And we've been interested in the major source of calcium signals in astrocytes, that is the endoplasmic reticulum. That you can see here on this micrograph, and here in blue in this 3D reconstruction from electron microscopy. And electron microscopy is the only tool for now that allows to have quantification of ER morphology and distribution in astrocytes, especially in astrocytic processes because of their small size. And it has been reported that around 47% of the processes that contact synapses contain endoplasmic reticulum, which means that actually more than half do not contain any. And interestingly, even within those that do contact endoplasmic reticulum, its distribution varies drastically and also changes in disease states, so that we would like to understand better what is the effect of such diverse distribution of the endoplasmic reticulum on local calcium activity at the vicinity of synapses. To do so, because those processes are very small, we used stochastic spatially extended modeling approach to investigate how ER distribution impacts local calcium activity. So indeed, if you take one process that is 1 micron long and 200 nanometers in diameter, 100 nanomolars of calcium, which is believed to be the basal concentration of calcium in astrocytes, corresponds to only 2 or 3 ions diffusing in this process, which leads to highly stochastic molecular reactions. And we have used the model that we have published in 2019 plus computational biology, and the modeled reactions are represented here. Basically, calcium influx results from influx from the extracellular space as well as from leak from the endoplasmic reticulum. And when both calcium and IP3 bind to the activating sites on the IP3 receptor on the membrane of the endoplasmic reticulum, which is a calcium channel, this leads to the opening of the channel and thus to influx of calcium from the endoplasmic reticulum into the cytosol. And then calcium is pumped back to the endoplasmic reticulum and the extracellular space, which allows the restoration of basal calcium concentration. We also take into account the fact that IP3 is synthesized in a calcium-dependent manner by PLC activity. In order to investigate the effects of ER distribution on calcium activity, we needed a spatialized model. And we have used the voxel-based simulation software developed by our team here in Okinawa that is called STEPS. So then in order to investigate the effects of the distance of the astrocytic endoplasmic reticulum to the synapse, we have created some simple geometries modeling one single astrocytic process that are characterized by diverse distances between the astrocytic endoplasmic reticulum and the synapse. To do so, we did not model the whole synapse. What we modeled is how neuronal stimulation affects concentration of IP3 within the astrocyte. So when neurotransmitters are released by active neurons, they bind receptors on the membrane of the astrocytes, which leads to the synthesis of IP3. And so we modeled neuronal stimulation directly with local increase of IP3 concentration at the tip of the process in all of those different geometries. And on this slide, I'm going to show you spatio-temporal traces of calcium activity depending on this ER-synapse distance, also called lambda. And you can see already here that depending on this distance, the calcium activity differs drastically. Notably, the activity seems to be larger when the distance between the stimulation site and the endoplasmic reticulum is smaller. And please note here that we are not plotting free calcium ion concentration in order to be closer to what is being performed experimentally. So in experiments, we cannot directly track calcium. So what is being done is that we use fluorescent indicators that fluoresce once bind to calcium. So this is an attempt to be closer to what is measured experimentally. And you can see here some representative traces in the whole process depending on the distance between the astrocytic ER and the synapse lambda. And you can see that as soon as that calcium activity decreases drastically as soon as this distance increases. We also performed some simulations in which there were no indicators added to the cell and measuring directly free calcium concentration. And you can see that, which would be what happens in the cell when there's no measurement from the experimentalist using fluorescent dyes. And you can see here that we still have a decrease of calcium activity with ER synapse distance. And here is a bit more quantification about those observations. And basically, calcium peak amplitude, duration, and frequency decrease when the distance between the synapse and the endoplasmic reticulum in the astrocyte increases. However, in those geometries in which the distance between the ER and the synapse increases, the overall length of the process also varied so that the cytosolic volume varied. And we were wondering to what extent this variation of cytosolic volume depending on the distance between the synapse and the ER was the cause of this decreased calcium activity when the ER is further away from the synapse. To test this hypothesis, we have performed some simulations in the exact same geometry every time, so 2 microns in length, but in which we varied the site of IP3 infusion at stimulation time. That was characterized by a diverse distance to the closest triangle on the ER surface, and that varied between 3 nanometers to 1.01 micrometer. And you can see here some representative CAG comps, so calcium bound to fluorescent indicator, or free calcium in the model that doesn't have any indicator. And you can see that there doesn't seem to be any significant difference. And here are the quantified data. And indeed, if volume stays the same, then there is no significant effect of ER synapse distance on calcium peak amplitude, duration, and frequency. So to conclude, we showed that the distribution of calcium stores, such as the endoplasmic reticulum, strongly impacts the spatio-temporal properties of calcium signals, especially when considering such small processes at the vicinity of synapses. Moreover, we showed that this effect is controlled by the variation of the volume of the processes in which the ER is distributed. Those results further highlight the need for characterization of the astrocytic ER morphology and distribution in live tissue, both in health and disease, so that we can have more refined models and provide better predictions of calcium activity and how it will impact neuronal astrocyte communication. And we are starting to work towards this direction, although using some data from electron microscopy, as no data to date is available on ER morphology in live tissue at this spatial scale. So here you can see a process of around 1 micron long again, reconstructed from electron microscopy by our collaborator, so-calle, and you can see the endoplasmic reticulum within it. And we're performing simulations in which we're changing the distribution of the calcium channels onto the membrane of the ER, as well as the distribution of the endoplasmic reticulum within the process, and investigating how this will impact microdomain calcium activity. So with this, I would like to thank my team members, especially my supervisor, Eric de Schutter, as well as Activiry, my collaborator and also PhD supervisor, and so-calle, our collaborator that performed all of the electron microscopy work. I would like to thank my funding, OIST and JSPS, and I would like to thank you for your attention. And if you have any questions, I would be glad to answer them. Thank you.